Welcome to the J and Margie Grosfeld Research Committee session. I'm one of the two moderators. My name is Agostino Pierro. I'm from the Hospital for the Children in Toronto, Canada.
And I'm Dr. Sean Kunisaki from the Johns Hopkins Children's Center in Baltimore, Maryland.
Both Dr. Kunisaki and myself are the chairs of the APSA Research Committee, and we are delighted today to have three exceptional presentations in this scientific section. Before starting, I would like just to remind you that this is the J and Margie Grosfeld scientific session on stem cells and tissue engineering. I know that everybody knows Dr. Grosfeld, and I want just to remind a few words about who Dr. Grosfeld was. He was one of the giants of pediatric surgery. And just to tell you, in 1972, he was appointed as professor director of the pediatric surgery at Indiana University and Riley Children's Hospital until his retirement, which occurred in 2003. Some important facts. In 1985, he was appointed chairman of the Department of Surgery, and he was the first pediatric surgeon in the States to be honored with this appointment. He has been an outstanding clinician and teacher, a master surgeon, and he was also very well known because he was the editor-in-chief for Journal of Pediatric Surgery, and he actually created also seminars in pediatric surgery, and was a senior author also in the two edition of the book chapters in 2 volumes that you are well known on pediatric surgery. For several generations, he's been really a mentor and a role model for pediatric surgeons, including myself. Important facts also, he was the president of this association, chairman of the surgical section of the American Academy of Pediatrics. In 2013, he was the vice president of the American College of Surgeons. He was also recipient of the most important honor, like the Lads Medal of the American Academy of Pediatrics, the Sir Dennis Brown Medal from the British Association of Pediatric Surgeons, and the Fritz Rebbein Medal from the UPSA, the European Association. Unfortunately, Dr. Grosfeld passed away, but both Dr. Grosfeld and Margie, his fantastic wife, have been supporting APSA and APSA Foundation for many years. And this research session is dedicated to Jay and Margie Grosfeld. So I think it's a great honor for us to do that. The section includes 3 important research papers, which covers different aspects of research related to stem cells and tissue engineering. One on the esophagus, another one looking at intestinal lengthening, and the other one on liver fibrosis and possible biliary atresia.
Our first abstract in this session will be presented by Mr. Jensen from Connecticut Children's Hospital. His presentation is entitled Comparison of Mesenchymal Stem Cells from Pediatric Control and Esophageal Atresia Patients for Use in Regenerative Medicine. Mr. Jensen.
Hello, my name is Todd Jensen. I am from UConn Health and Connecticut Children's. I'd like to thank the organizers for the opportunity to present our research. I will be talking about a comparison of mesenchymal stem cells from pediatric control and esophageal atresia patients for use in regenerative medicine. Previous studies had been reported in 2018 by a small biotech company called BioStage. We currently have an academic partnership with this biotech company and have been working with them for the last 3 to 5 years. They published on their new combination device that utilizes a patient's own stem cells that are then seeded onto a tubular scaffold that is manufactured. The goal of this device is to promote esophageal regeneration and bridge esophageal gaps seen in adult cancer patients. This was conducted in a very large preclinical study using adult pigs and demonstrated within the first couple of months after surgery that continuity was restored and tissue was regenerated. A similar study was conducted with this biotech company in our research group using a piglet model of esophageal loss. In this case, amniotic fluid-derived stem cells were used in a small tubular-sized scaffold. The same results were seen, and by the end of the study at 6 months, The piglets were flourishing and almost adult size. In addition to this exciting data, the first in-human case was documented in the beginning of 2019. Through compassionate use, this device was used on a male patient who presented with a very large cancerous mass in his chest, part of which had compromised the esophagus. This was implanted into that region that was resected, and we are happy to report that the tissue did indeed regenerate. However, the patient ultimately died from complications from other procedures. Therefore, to determine whether this technology would be applicable to the pediatric population, we obtained fat from pediatric patients presenting with various types of esophageal atresia at Connecticut Children's. We also utilized patients undergoing bariatric surgery at Connecticut Children's as control patients. The goal was to isolate and expand adipose-derived mesenchymal stem cells from these patient samples and characterize them, and then seed them onto these tubular scaffolds to ensure that they would act as we had similarly seen in the pig cells. Here is a table illustrating the patients that were enrolled in the study and the various categories that we had placed them into. First, we characterized the cells by flow cytometry, and as you can see by the graph on the left side of your screen, the majority of the cells expressed mesenchymal stem cell markers CD90, CD105, CD73, and CD44, and did not express hematopoietic stem cell markers CD34, CD45, or HLA-DR. Gene expression analysis also confirmed similar findings, as well as the expression of cytokines IL-6, IL-8, and IL-10. Cells from these patients were expanded and seeded onto these tubular scaffolds that are mounted in disposable bioreactors, as you see in the picture below. Over the course of 6 days in this bioreactor, they rotated at 1 revolution per minute, and media was analyzed and changed every 2 days. As you can see by the graphs here, glucose levels decreased, indicating metabolic activity by the cells, as well as lactate levels increasing. At the end of 6 days, portions of the scaffold were stained with live dead staining, and as you can see here indicated by green staining, all of these cells are still viable at day 6. Portions of the scaffold were also analyzed for gene expression and compared to the cells that were initially seeded on the scaffold. There was significant upregulation in IL-6 expression from the patient samples, normal and TEF patient samples, but this was not the same finding in the long gap esophageal atresia samples. This may be important as IL-6 is a key cytokine in wound healing, but further studies are needed to determine that. What's important to note and conclude from this study is that cell lines were derived from as little as 65 milligrams of fat, which provides feasibility of translating this technology to small babies who undergo procedures in the first couple of days of life. There were some genotypic differences between control esophageal atresia and long gap esophageal atresia patients as expected, and they were able to all be cultured on synthetic scaffolds for 6 days with metabolic activity noted. This combination device has been approved by the FDA just recently for clinical trials and will soon commence in the next 6 months for the adult indications. This technology was developed by the BioStage team seen on the right side of your screen. And on the left side of the screen are all of the researchers at Connecticut Children's who helped with this project. Funding was also provided by the NIH through the NICHD for a small business grant, phase 1, phase 2 application. And I will take any questions, and here is my contact information.
Thank you, Mr. Jensen. The invited discussant for this paper is Professor Paolo Di Caprio from Great Ormond Street Hospital in London, UK.
Thank you. Thank you for the opportunity to be here discussing this interesting paper. Just a few questions, Mr. Jensen, if I can. You have nicely shown that actually mesenchymal stem cells coming from different sources and for children affected that may benefit in the future may have similar power to the one coming from control. But there's quite a difference in terms of age of the two groups that you taken in consideration. Do you think this may affect your study?
Absolutely. So it's been well described in the literature that the age of MSCs does affect their potency and in some cases the cytokines that are produced. So our aim was to try to get younger patients for the study. However, the most easily attained for this study was the bariatric patients. It just so happened to be that these were considerably older, but we do believe a lot of the differences we are seeing could be age-related.
And do you think before you'll be able to translate this, you will need at least to validate in a population of children that you could use as control in terms of seeing really if the potential of your cells will be adequate to complete your study then clinically?
Absolutely. I do believe that in order to ensure that the cell types that we obtain from the long gap and esophageal atresia patients are similar to that of children the same age, we really do need to have those controls, and we're actually in the process of getting those controls now through patient consent.
Dr. Fink, our group has shown in various studies that this is a real possibility for the future of treatment of these devastating diseases. Is just a couple of questions around the approach. I can believe that when you fill a gap with a stent material, the epithelium will grow over that gap and function as a breach. But it's a bit more difficult to understand how the muscle may grow. And of course, to the regeneration of the esophagus, the muscle function is extremely important. You're using mesenchymal stem cells, which we know they can regenerate adipose or bone tissue. But the question around the muscle is a bit more a difficult one. What is your feeling having worked in this field now for quite some time?
So it's a very good question. Thank you. So we believe really the mechanism at play is really paracrine. And what we have shown in those animal models is that the initial cells that populate around the scaffold that is then extruded do appear to be muscle or fibrotic in nature. Basically, what we have shown, at least by 365 days in our animal studies, is that there's some degree of peristalsis. But when we look at cell tracking studies and where these cells actually go once we implant them into the animals, looking at GFP tracking studies, they end up in the perivascular space, but only a few do. Most of them are cleared by the time the scaffold is actually removed. So it brings up an interesting point of the role of these cells and whether it's simply paracrine or whether they actually play a structural part.
One last question, if I may, regards the vascular supply. So we have had previous work done on translational of the trachea. So when we have replaced trachea segment using an engineered trachea, we had to use the omentum as a vascular supply to the trachea. And we know that cartilage requires much less vascular supply than muscle. How do you think that you can actually offer that vascular supply to your patch, being the esophagus and having to fill probably a few centimeters of gap to be efficient?
So what we've actually noticed is that in our animal studies that, believe it or not, implanting these segments that are about 5 centimeters in length into the thoracic region, we've used CT scan and actually have been able to show that there's been branching off the descending aorta that have actually led to vasculature of this graft. The best we can hypothesize is that due to the amount of VEGF that is expressed by these cells that are on the scaffold, that this seems to drive the angiogenesis by the vessels that are close by.
Thank you, Professor Di Caprio, for your astute comments. Mr. Jensen, I also had one other question for you. I was wondering if you could tell us a little bit more about the polyurethane scaffold itself. My understanding is that it's a synthetic nanofiber-based scaffold. And so with all these synthetic scaffolds, do you know anything about the biocompatibility of this when implanted and what its long-term degradation characteristics are in vivo?
That's a great question. So the company that we collaborate with doing this work manufactures this scaffold. And as you said, it's polyurethane, it's nanofiber spun, then it's plasma treated so that the cells do indeed attach. Well, this scaffold has been put through a variety of testing. The most important thing I can underscore is that given the model that we've used, this scaffold actually is extruded with the first stent at day 21. So the nice thing is within the first 21 days, it does not integrate. There is no degradation shown. The tensile strengths and burst pressures of that scaffold are actually as it was when it was put in. And so given that they're really, and especially in the leachable studies and other things done in the lab, there's nothing really that shows that there's any compatibility issues. And the part here that's kind of most exciting is that we don't really have to worry past the first 3 weeks about this scaffold still being in place.
Fascinating, excellent work, and congratulations again.
Now we move to the next presentation in the program, and the title is Mesenteric neuroscularization during spring-mediated intestinal lengthening. And the presenting author is Dr. Modupeola Diallo from...
Hello, my name is Modupeola Diallo, and I'm a postdoctoral fellow at Dr. James Dunn's lab at Stanford University. I will be presenting mesenteric neovascularization during spring-mediated intestinal lengthening. I have no disclosures. Short-gut syndrome is a gastrointestinal disorder characterized by malabsorption and malnutrition secondary to intestinal loss. In the pediatric population, the most common causes are neck, intestinal atresia, and midgut volvulus, which can lead to an extensive intestinal resection. Treatment for short gut is largely supportive, and although lengthening procedures do exist, these treatments can be morbid. Our lab has focused on spring-mediated distraction enterogenesis, in which nitinol springs are compressed, encapsulated, gel-coated, and inserted into the jejunum of female pigs as a way to lengthen the bowel. Previously, we have demonstrated 1 to 3-fold intestinal lengthening with spring constants as low as 2 newtons per meter and as high as 21. Throughout these experiments, we have also noted changes in the mucosa and muscularis layers in our spring segments of small bowel in response to the lengthening process. As early as postoperative day 7, we can see an increase in the thickness of the muscularis and non-muscularis layers of jejunum as compared to normal bowel. Our current hypothesis is that in order to support this growth, there's an increase in the mesenteric vasculature in order to supply blood and nutrients to growing bowel. Female juvenile Yucatan pigs received a midline laparotomy and were implanted with gel-coated, encapsulated, compressed nitinol springs. Intraoperatively, the number of mesenteric vessels were noted at the spring segment and the control segment. Postoperatively, the pigs were placed on an all-liquid diet. They were sacked on post-op day 7 and 14, at which time the number of mesenteric vessels were recounted. H&E staining and immunohistochemistry with smooth muscle antibody was performed on both the control and spring segments. All pigs survived to their respective date of sack. On average, we saw a 3-fold increase in the spring-lengthened segments of bowel. On a typical segment of bowel where the spring was implanted, there was an average of 2 macroscopic mesenteric blood vessels. After lengthening, we saw a statistically significant increase in macroscopic blood vessels, where an average of 4 vessels were noted. The number of vessels on the control bowel remained similar. On H&E staining and immunohistochemistry, we noted the extent of microscopic blood vessels. On the y-axis, we have the number of vessels, and on the x-axis, the segment of bowel. The control bowel had an average of 2 arterioles and venules. When compared to the spring-lengthened segments, there was a statistically significant increase in microscopic blood vessels, with an average of 3 arterioles and 4 venules seen. The density of mesenteric vasculature was also calculated. On the control bowel, there was an average of 3 arterioles and 4 venules per millimeter of segment of mesentery. After lengthening, there were 4 arterioles and 8 venules per millimeter of mesentery, which was also statistically significant. In conclusion, increased vasculature in the mesentery is seen in lengthened segments of bowel, likely due to the increase in demand of blood supply and nutrients to support growing tissue. Further studies are needed to determine the pathophysiology of the angiogenesis in terms of if there are particular growth factors that are responsible. Thank you.
Thank you very much for your wonderful presentation. The discussant for this paper is Dr. Greek Wright from Children's Hospital Los Angeles. He's a famous pediatric surgeon and even more famous scientist with a laboratory which is focused on regenerative medicine and tissue engineering.
So I've been looking forward to discussing your paper with you because it's beautiful to see that lengthening of the intestine and native intestine so that we don't have to worry about building it up from the ground up, which is obviously a very big problem and quite expensive to do. So this could be a beautiful approach to help many children and could actually reach a lot of people. As I was looking through the old papers from your lab, it struck me that I don't actually understand the way that the spring unfurls itself. So I know that it's encapsulated and it's implanted that way, and then it lengthens, and the post-operative pictures are beautiful, showing that over time it really has lengthened and generated more intestine. But my first question was just if you could walk me through sort of, I know you only harvest when you harvest, but do you have an idea of how quickly it opens itself up and assumes that final composition?
Sure, thank you for your question. So when we implant these springs, they are encapsulated and the capsule degrades actually pretty quickly. We've noticed in previous models, surgeries where we've done as soon as post-op day 3, that the capsule has already disintegrated.
And so then I imagine you kept the animals on the clear liquid diet so that things can flow through the spring. And then I see that it really has opened up tremendously. And then you were able to show that there was much more vascularity to the area where you had put the spring, which was beautiful and sort of builds on what we were just speaking about in the last presentation, that there might be some VEGF and other factors that are leading to that. So I immediately wondered about the cells inside of the intestine, and does the epithelium seem as though you have deeper crypts and taller villi, or do you have a sense if the stem cells or other parts of the intestine that'll help you in your attempt to get more intestinal surface area are coming along for the ride with the larger size of the intestine?
So we do notice within our spring segments that the crypts are a little bit deeper, and we've also seen that the villi are a little bit thinner, especially in the models where we've kept for up to 3 weeks. See these structural changes within the bowel, and then within the layers of the intestine itself, we do see some inflammatory cells within the spring lengthened segments, also within the segments that are proximal and distal to the spring. Over time, we do see some restructuring of these mucosal layers. We see some thickening in some of the layers, and that sort of stabilizes throughout the time that the spring is within the bowel up to 3 weeks.
I think as a pediatric surgeon, we've all been confronted with where you have an atresia or some other obstruction, and you get a 3-to-1, a 4-to-1, even more of a size discrepancy, and you start to worry about the function of the very distended segment. So I also wondered if you're starting to have any insight into the motility through that banded segment, because it's wonderful to have more, but then sometimes it isn't once it gets too sort of big and baggy.
In terms of motility, intraoperatively, when we do sac our pigs, we do see some gross movement within the bowel. We're still within the process of determining how robust that contraction still is. And then in the future, we're also looking to see how well these lengthened segments absorb the nutrients that pig or in soon to be a child will do, how well it will do.
Well, it sounds like you're starting to cover all the steps that you need to walk toward a clinical trial. So I hope that's in your plans because it's very promising.
But Paola, this is fantastic. And thank you, Tracy, for your comments. Can I just go back to what Dr. Grosfeld just said at the end? Can you tell us a little bit more how far you think you will be from a clinical application?
So currently we are testing a spring-loaded device within our porcine model. The device has received a humanitarian use designation by the FDA, and we will file an application for the FDA to do a first in-human clinical trial. The clinical application will really occur when the device is approved by the FDA, which is the most current hurdle. And we do see some potential difficulties getting enough funding to commercialize the device and potential morbidities associated with the device. We're hoping to do a couple more experiments within our porcine model with the spring-loaded device before we move on to the second step of in-human trial.
I would like really to congratulate you for this fantastic work, and congratulations also to Dr. Dan for the work in his lab. Thank you.
Thank you so much.
Our final abstract in this session will be presented by Dr. Chusi from the Hospital for Sick Children in Toronto. Her presentation is entitled "The Fibrogenic Injury of Liver Ductal Organoids is Rescued by Human Amniotic Fluid Stem Cells." Dr. Chusi.
Hello everyone, my name is Sinobon Chusin. I'm pediatric surgeon from Thailand and now currently working as a research fellow at Dr. Pillow Lab, the Hospital for Sick Children in Canada. Today, I would like to present my project about the fibrogenic injury model in liver ductal organoids and the role of human amniotic fluid stem cell in rescuing the injury. Liver fibrosis is a wound healing process present after chronic liver injury. This process is characterized by accumulation of extracellular matrix and collagen in liver. Although this ongoing process is reversible, it can progress to liver cirrhosis and end up with end-stage liver disease, which increases morbidity, mortality, and requirement of liver transplantation. Understanding the mechanism of liver fibrogenesis is essential to develop a novel therapy for preventing or reversing liver fibrosis and improve clinical outcome. In this process, the fibrogenic cytokine, including transforming growth factor beta-1 and platelet-derived growth factor BB, are released from the injured liver cells and initiate fibrogenesis process by activating hepatic stellate cells, which are the major population of cells that produce collagen in liver. Cholangiocyte apoptosis is one type of cholangiocyte injury that has great influence on the pathogenesis of cholangiopathic disease, such as biliary atresia and primary sclerosing cholangitis. The apoptotic cholangiocyte interact with itself or other cells in liver through the inflammatory cytokine and fibrogenic cytokines. This results in sustentation of biliary inflammation and fibrosis. It has been demonstrated that the liver of biliary atresia patients has increased number of apoptotic cholangiocytes. However, the interplay between cholangiocyte apoptosis and the progression of liver fibrosis is not well characterized. In this study, we hypothesized that the cholangiocyte apoptosis is also involved in liver fibrogenesis by releasing fibrogenic cytokines. Amniotic fluid stem cells are multipotent cells present in amniotic fluid. It has been described as a novel cell source for stem cell therapy due to their potential of high differentiation and proliferation. Moreover, these cells can be collected and expanded for subsequent application in perinatal period. Various studies have demonstrated that amniotic fluid stem cells can reduce inflammation, inhibit cell apoptosis, and stimulate tissue regeneration through their paracrine effect. We hypothesized that the capacity in minimizing tissue injury of amniotic fluid stem cells can be applicable to treat liver fibrosis by a beneficial reduction of the injury-induced fibrogenesis. In this study, we aim to investigate whether human amniotic fluid stem cells have an anti-fibrogenic effect in the injured liver ductal organoids in terms of reducing fibrogenic cytokine secretion and reducing cholangiocyte apoptosis. Liver ductal organoids are in vitro self-organizing 3D structures derived from liver ductal progenitor cells. These organoids express both biliary progenitor marker, cytokeratin 7, and hepatic progenitor marker, alpha-fetoprotein, so they have the potential to differentiate into both cholangiocytes and hepatocytes. Moreover, they are capable of self-renewal and recapitulate the functionality of in vivo bile ducts, which allow us to develop a disease model of bile duct disease. In our study, liver ductal organoids were generated from the liver of healthy mouse pup. Intrahepatic bile duct fragments were isolated by using tissue dissociation cocktail. Then the drug fragments were cultured in multi-gel dome and supplemented with culture medium to allow the forming of the 3D structures. In treatment group, amniotic-free stem cell purchased from cell portion were co-cultured with the drug fragments and maintained in the gel dome before an injury induction. We established an injury model by using acetaminophen, which is a hepatotoxicity agent that induces liver cell apoptosis. The injury was induced in both injury group and the stem cell treatment group on day 4 of culture and maintained for 24 hours. Then the organoids were collected for further analysis. After injury, the morphological injury of the organoids could be obviously detected, as shown by increased darkened cell or the dead cell inside the organoids. The organoids which were co-cultured with amniotic-free stem cells appear to have less injured cells, as shown by decreasing of dark area in the organoids. We also found that the injured organoids significantly increased expression of fibrogenic cytokines, TGF-beta1 and PDGF-BB, and co-culture of amniotic-free stem cell with the organoids significantly decreased the expression of these cytokines. To evaluate the cholangiocyte apoptosis, liver ductal organoids were stained with cell apoptotic marker, CRISPR-Cas3, as shown in red color. We found that the injury induced by acetaminophen was associated with cholangiocyte apoptosis, as indicated by significantly increased number of CRISPR-Cas3 positive cells. And this apoptosis was significantly decreased by administration of amniotic-free stem cell. Cholangiocyte proliferation was also evaluated in the organoids by staining with proliferation marker Ki67, as shown in green color. The injury significantly decreased cholangiocyte proliferation, and interestingly, amniotic-free stem cells significantly enhanced the proliferation of cholangiocyte in these injured organoids. In summary, our injury model in liver ductal organoids demonstrated that after exposure to acetaminophen, the injured cholangiocytes underwent apoptosis and increased expression of potent fibrogenic cytokines. This finding indicates that apoptotic cholangiocyte might play a role in liver fibrogenesis by secreting fibrogenic cytokines. Co-culture of amniotic-free stem cell rescue cholangiocyte from the injury by decreasing cell apoptosis and promoting cell proliferation, which result in reduction of fibrogenic cytokine releasing. This result suggests that amniotic-free stem cell can protect bile ducts from injury, and this protective ability might lead to inhibition of further liver fibrogenesis. In conclusion, human amniotic-free stem cells have potential to attenuate liver fibrosis, especially in disease that involve bile duct injury. Further study in an animal model is needed to evaluate the efficacy and the translational application of amniotic-free stem cell in berry artesia. Thank you for your kind attention.
Thank you, Dr. Chusip. The invited discussant for this paper is Dr. Gail Besner, who's Chief of Pediatric Surgery from Nationwide Children's Hospital in Columbus, Ohio.
Thank you, Dr. Chisholm, for that very well-organized presentation and for sharing your important work from the Piera Laboratory with us today. It wasn't that long ago that it was believed that liver fibrosis was an irreversible process that could not be cured. However, over the years, this has been found not to be the case. Numerous animal studies have shown that fibrosis of the liver and many other organs can be blocked or reversed using agents that interfere with fibrogenic or inflammatory pathways, such as targeting of TGF-beta, which you just spoke about. More importantly, there's evidence from the clinical arena that liver fibrosis is in fact reversible. For example, a large proportion of patients on antiviral therapy for hepatitis show considerably reduced liver fibrosis as well as reduced viral load. This tells us that removing the primary cause can itself be sufficient for fibrosis to be cleared. However, there are still patients whose fibrosis persists even when the primary disease is successfully treated. And it's this group of patients for whom antifibrotic therapy is most desperately needed. The talk we just heard shows that the cholangiocyte is also a target for stem cell action and resolution of cellular injury. This is important because most other studies have addressed the interactions of stem cells with hepatocytes. Therefore, this work adds an important new dimension that points to the versatility of stem cells as antifibrotic tools in different liver diseases. With that said, I have a few questions for you, which I'll ask you one at a time. First, can you tell us a little more about your cholangiocyte model? Specifically, is a direct interaction between the stem cells and the organoids necessary for the stem cells to elicit their beneficial effects?
Thank you, Dr. Bensner, for these excellent questions. At this moment, we cannot tell if the interaction is direct or indirect. Recent studies have proposed that amniotic-free stem cells provide benefit in different disease models through a paracrine effect. For example, in an intestinal epithelial injury model of necrotizing enterocolitis, our group has found that the amniotic-free stem cells decrease inflammation by stimulating COX-2 pathway and also improve epithelial regeneration by upregulating Wnt signal. So we speculate that amniotic-free stem cells also work through the paracrine effect to minimize cholangiocyte injury in our model. Therefore, to confirm the indirect effect of amniotic-free stem cells, we plan to give the liver ductal organoid stem cell condition medium, which is the medium that contains factors secreted from stem cells during culture, and we will look for the results.
Thank you. In models of fibrosis, inflammation induces activation of stellate cells, which then leads to the fibrosis. In your model, does acetaminophen stimulate the production of inflammatory mediators by the organoids? And if so, is this attenuated by the stem cell therapy?
We have actually also done further experiment looking at the inflammation. We found that the expression of inflammatory cytokines, including interleukin-6, TNF-alpha, do not increase in our cholangiocyte model. Therefore, we think that the injury effect of acetaminophen is not related to inflammation, but it is a direct activation of fibrogenic response.
Oh, very interesting. Thank you. Lastly, stem cells have been shown to be highly anti-fibrogenic in many organs in vivo, including the liver. Do you think there are mechanistic differences in how stem cells work in your ex vivo cholangiocyte model compared to models of liver fibrosis in vivo?
The in vivo experimental study of carbon tetrachloride induced liver fibrosis by Sean Yupeng in 2014 has shown the anti-fibrogenic effect of amniotic-free stem cells, which decrease collagen deposition in liver. However, in the case of cholangiopathic disease, such as biliary atresia, we believe that liver fibrosis is a consequence from a fibrogenic cascade, which results from the early fibrogenic response of intrahepatic cholangiocytes after injury, and also from further fibrogenesis process of other liver cells. I would like to stress that, in fact, we are the first to create a cholangiocyte injury model to mimic the pathogenesis of biliary atresia, which involve cholangiocyte injury and apoptosis. And we believe that by using our model, we could develop a way to intervene the fibrogenic cascade at an earlier stage of liver fibrogenesis. We shown that amniotic-free stem cells reveal a apoptosis of cholangiocyte and the expression of fibrogenic cytokines. We conclude that amniotic-free stem cells may play different roles in different stages of liver fibrogenesis. Therefore, our results suggest that these stem cells provide beneficial effect in the earlier stage of liver fibrogenesis. And this could prevent the further progression into liver fibrosis or cirrhosis in biliary atresia. We hope to use this stem cell. If it's possible, we can give this stem cell while we're doing surgery, or after calcite surgery. And we hope that it could minimize the intrahepatic cholangiocyte injury and prevent further fibrogenesis process after surgery.
Thank you again for a really lovely, well-presented study.
Thank you very much.
Fantastic work. Thank you, Dr. Chusip and Dr. Besner, for those insightful comments and great discussion. I did have one final question for you, Dr. Chusip. Unless I missed something, it seemed like the model that you proposed was really more of a prevention model rather than a therapeutic model, and that the injury was basically given after the stem cell administration. Have you done further work on that to see if this could be used more as a therapy? Because I think that would be really exciting in terms of using these amniotic fluid stem cells.
Thank you, Dr. Kunisaki, for an excellent question. However, we didn't explore yet about how we can give this stem cell in animal model, or we haven't tested yet about in the biliary atresia model, but we have planned to test this stem cell in biliary atresia model.
Fantastic. Thank you, Dr. Chusip, and congratulations for some really nice and elegant studies.
Thank you. This concludes our brilliant session on the stem cells and tissue engineering research session. I would like to thank all the presenters for these very nice presentations, the expert discussant, which contributed with their comments and questions. Sean, do you have anything else to add?
No, I think this was a great, fantastic session. This is certainly one of my favorite topics in pediatric surgery. I think this also is a tribute to really the importance of surgeons in basic science. As most of you know, and I'm a little biased, of course, this entire field of tissue engineering and regenerative medicine has really had its origins in pediatric surgery with Dr. Vicanti, our past APSA president. So I'm really excited to talk about some of the work that's been going on over the last 25 years and what currently is the status today. And so, I love it. And this has been really exciting for me to be involved with this.
So I think Dr. Grosfeld will be proud of all these presentations. And I'm sure that our president of APSA would be very pleased to see what has been shown today. Thank you.